Today I will be discussing current understandings surrounding iron metabolism and its relation to the devastating neurodegenerative disease called dementia. Although various studies exist linking iron regulation to neurodegeneration, further literature is required to accurately explain this phenomenon. Dementia is an umbrella term used to describe a variety of symptoms characterized by progressive cognitive decline, which negatively affects an individual's ability to function normally in aspects such as memory, language, and physical capacity. There are multiple sources of dementia, each having their own unique symptoms and causes, however share some overall similarities such as short-term memory loss, personality changes, and loss of bodily function in the later stages of the disease. The most common of these causes being Alzheimer's disease, making up 60 to 80% of all dementia cases, followed by vascular dementia, Lewy body dementia, and frontotemporal dementia. According to the National Bureau of Statistics 2018, dementia was the second leading cause of death in Australia, costing the Australian government over $15 billion. As the Australian and worldwide populations age, the prevalence and cost of dementia is increasing at an exponential rate, and these numbers are expected to continue to rise as such without a medical breakthrough. Currently in Australia, there is an estimated 459,000 people living with dementia. Alarmingly, this number is expected to rise to 590,000 in 2028 and 1,076,000 by 2058. Therefore, knowledge surrounding the potential causes of dementia, such as the role of iron metabolism, must be further researched to break this current trend. Iron is the most abundant metal in the body. It is responsible for a variety of cellular mechanisms such as the transport of oxygen, neurotransmitter synthesis, myelination of neurons, and mitochondrial function. The body has specialized mechanisms to thoroughly regulate its levels as a means to ensure homeostasis, as both excess iron and iron deficiency are associated with neurodegeneration and cognitive decline. Within the food we eat, there are two types of iron, absorptive heme iron Fe2+, and non-absorptive non-heme iron Fe3+. The more easily absorbed heme iron is found in animal products such as meat, chicken, and fish, whereas non-heme iron is found in plant foods such as vegetables and cereals. This ingested iron travels through the digestive system until it reaches the duodenum of the small intestine, where it is selectively absorbed by the duodenal cells according to the iron needs of the body. From here, it either binds with apoferritin to be stored in non-toxic form ferritin, or travels through circulation in its transportable form transferrin. Transferrin is used to transport iron around the body to organs which require its uptake for their own metabolic needs. However, unlike other organs, the brain cannot uptake iron directly from the circulatory system as it rests behind a vascular barrier called the blood-brain barrier. The blood-brain barrier consists of capillaries that hold endothelial cells wedged extremely close together, forming tight junctions. These tight junctions are used to prevent the passive diffusion of unwanted proteins and metabolites from the bloodstream entering the brain. Transferrin receptors are situated on the endothelium, in which they receive transferrin and mediate its release into the brain. However, issues arise in these complex physiological processes as the human body and brain age. Recent studies such as Hoskins et al.'s 2018 systematic review on iron metabolism and cognitive decline suggest that iron regulation within the brain becomes much more difficult as we age, ultimately resulting in either accumulation of iron or lack of iron in the brain. The accumulation of iron in the brain, or hemochromatosis, has been extensively linked to neurological and cognitive decline. This increased concentration of iron with aging is suspected to be caused by several factors, including increased blood-brain barrier permeability, inflammation, redistribution of iron within the brain, and changes in iron homeostasis. This notion is further expressed in Erdos et al.'s 2017 article, as they suggest increased vascular permeability is a primary cause of high iron deposits in the brain. This means, as the brain ages, the effectiveness of the blood-brain barrier decreases, resulting in an increased amount of iron allowed to enter and deposit inside the brain. But why is this an issue? As we know, high levels of iron can be toxic and cause oxidative damage. There are three main mechanisms surrounding why high iron levels within the brain subject individuals to a greater risk of developing dementia. Abnormal iron metabolism and high blood iron levels have been shown to generate highly toxic hydroxyl free radicals through the reaction of ferrous iron with hydrogen peroxide. This phenomenon is known as the Fenton reaction. According to Zhao's 2019 article, this reaction can cause oxidative damage to lipids, proteins, and DNA, ultimately leading to cell death within the brain. Moreover, high levels of iron have been shown to interact with beta amyloid to form plaques and with tau proteins to form neurofibrillary tangles. This is alarming as plaques work to obstruct the transmission of brain cell signals, whereas tangles kill them. Together, all these factors promote the development of Alzheimer's and dementia, as they all induce oxidative stress and cell death. This is further exemplified by post-mortem studies of Alzheimer's patients' brains. These studies demonstrate that Alzheimer's sufferers have high levels of iron deposits, beta amyloid plaques, and tau tangles in their brains, raising questions about the possible solutions to this issue and guiding the basis of further research. Now on to low iron levels in dementia. Physiologically, one of the main roles of iron in the body encompasses the transport of oxygen. 70% of the body's iron is found within red blood cells in the form of hemoglobin. Hemoglobin binds with four oxygen molecules and circulates the bloodstream, delivering this oxygen to the body's cells and tissues. When iron levels are low or the individual is anemic, the effectiveness of oxygen transport is disrupted, and the body is deprived of its much needed oxygen. Even for a short period of time, lack of oxygen to the brain can cause irreversible damage. According to Hong et al.'s 2013 study, this deprivation of blood or oxygen flow to the brain may result in illnesses such as vascular dementia. So what can we take away from this knowledge? From this research, it can be derived that iron metabolism plays quite a large role in the development of dementia. Future research is primarily focused on the prevention, early detection, or slowing disease progression. Treatment methods such as the use of iron chelators such as lipoic acid or lactoferrin have been shown to lower iron levels in the brain as they are able to cross the blood-brain barrier. These candidate drugs, once inside the brain, bind to iron, reducing iron levels, thus mediating the symptoms associated with dementia. Nevertheless, more research is required to uncover more effective iron chelators and completely understand their mechanism within the brain. All in all, higher quality and more comprehensive studies are needed to fill in the gaps in the current evidence base and obtain a greater understanding of the role of iron in dementia and